But okay, individual genes are important for different stuff, but actually there are just a few genes which we can tell like confidently that they're indeed important, even if you find changes in these single genes, like top important genes. Genes are related with each other. They interact, they form gene nets. So if you wanna really discover whether this trait is about you or not, you should analyze like a whole set of your genes. And for doing that, we use polygenic risk scores. And I am happy to give talk to my colleague who will tell about polygenic risk scores.
Hi, everyone. My name is Alina, and I will talk to you about polygenic risk scores. And I'll just share my screen. So thank you, Olga, for a nice introduction. And yeah, now we have already know how we can work with single polymorphism, but it's probably not enough just read the descriptions of genes or polymorphism. You need to have some model that can predict phenotypes. The first thing that you can presume, okay, I just get the coolest neural network possible, the best architecture, and you get some interesting predictions. But when you start dig data, you realize, firstly, it's extremely hard to get access to that data, in biobanks. Everything is so bureaucratized. And secondly, that you discover that this data can occupy a lot of gigabytes, but the number of samples are usually smaller than the number of features. So how you can see in this slide, you can construct some matrix. In UK Biobank, it's about 500,000 biosamples. In Japan Biobank, 200,000 samples. And features, it's about 850,000 variants. So you have matrix where the samples as rows and features as columns, and you can make some prediction where there is health, where there are disease.
Qu'est-ce qu'elle a dit?
Elle a remis Cohen en place l'autre jour. Hein. Il a couplé en plein milieu de phrase. Mais il dit, laissez-moi terminer. Virgule.
Okay. And but this matrix is balanced in terms of number of samples and number of features. And it's extremely easy for model to overfit. So overfitting is when we have good performance on the training data and poor generalization to other data. So, and another problem that we have far more variance than we directly measured. So it's towards 90 million variants imputed. So imputation is a kind of statistical inference of unobserved variants. We do it by comparison with the reference genome. So why we want to predict something, why we want to build some machine learning models. With that models, we can predict complex traits. And with that kind of models, we can facilitate disease screening and prevention at the population scale, aid in the development of personalized medicine, improve symptom diagnosis and intervention at an early stage of diseases, We can reconstruct the genetic predisposition of an individual to a particular disease, and also we can apply it to the breeding values of animals at some kind of genomic selection. And what is polygenic risk score is an estimate of an individual's genetic risk for some trait obtained by aggregating and quantifying the effect of many common variants. So we can see this simple formula. We just summon up some product of beta is the effect size of variant. We can just get it from GWAS analysis, some statistical analysis of your data. Multiply by dosage, number of copies of SNP and the genotype of individual. How we can calculate dosage? It depends of the method you use to infer your SNP. So if you directly measure it, SNP will have probability 1 of one genotype and 0 for the others. But at an imputed SNP, an individual might have, for example, a 90% probability of being homozygous for allele A and 10% of probability of being heterozygous. So how we can turn from this simple formula, which we already know for PRS, to machine learning models? Just through linear regression. We all probably know this simple formula from our school years. It's just linear regression, where y is an in vector of the phenotypes. So left part is phenotypes and right part of equation is genotypes. X as an n by p matrix of genotypes collected across p's and p's on the same set of individuals. Beta is a p vector of SNP effect sizes, and epsilon is an n vector of residual errors. Despite its simplicity, the above model is instrumental for understanding almost all existing PRS methods. But we can keep in mind that we have unbalanced data, so we can solve this problem with applying some kind of regularization to our data. So it's just trying to shrink coefficients in equation to 0. And how can we actually predict new observations through PRS construction? We can predict phenotypes for newly observed individuals using the estimated SNP effect size beta from the modeling equation. Specifically, once we have obtained the p vector of genotypes XL for a new individual L, we can simply plug in the SNP effect estimates to obtain the predicted phenotype value, PGS, as in that way on the bottom of the slide. And we can explore some existing models. So some scientists have already overcome those difficulties with bureaucratized biobanks and with overfitting models, and successfully applied some models to the data. And we can see that some model with regularization, lasso, elastic net is the most efficient models. We can understand it just by R square coefficient is a determination coefficient, which tells us how good is the model. So as closer to the 1, as the better the model. So when we explore our own genome, we can find some polygenic risk score. We can find it free on the internet. There are 2 ways. We can just find paper for topic we're interested in, or just find hosted and curated scores, for example, Polygenic score catalog, PGS catalog, and cancer PRS web. They're very convenient way to find PRS and information from the article from the sample or about the samples. And about some specifics of PRS, we should keep in mind that the larger datasets give more power to detect association of SNP with the trait of interest and have been shown to yield scores with higher predictive capability. And the PRS will perform best in population of the same ancestry as those in which they were derived. And finally, when using an existing PRS, it is important to first ensure that it is of good quality and is appropriate for the analysis data. We can find this information just in papers. And generally, we need to know what kind of information we need to find in our waiting files of polygenic rescores. At the minimum, the information needed to replicate the PRS is a list of SNP, so with some RSID or base pair positions on chromosome, the effect allele for each SNP, and the effect size for each SNP for the condition of interest, and the genome build. And I have pointed out that training models is extremely tough and bureaucratized. So far, we decided we can use already trained models PRS with published coefficients to provide output to our users about their disease risk, and Alex Karmazin will talk more in depth how we do it now. Thank you.
Hello, I'm Alex Karmazin. I'm software development for the just-dna-seq project, and now I will explain a little bit our architecture and how we use PRS with OpenCravat. So nowadays, there is plenty of PRS on the web. You could use this catalog and just download a PRS as a text file, and then you should do something with it to get results and helpful information. In our case, we use the PRS for the personalized reports that we developing now. So the general idea is that we have the PRS text file. We make a parser that gets this data and creates a SQLite database. Then we create OpenCravat or OCravat. It's the same annotator. It's just a piece of Python code that have some function that triggers by OpenCravat each time it has the new SNP, and we just make a query to our database, and if we have some data, we could just put this data in the table that we see on OpenCravat. In our case, these data will be the weights that have this PRS that we are using. And here we have some trick that we have different versions of the VCF file, and the PRS could be trained on one version, and our user could have some different version. So that's why it's easier to stick to such arrays that have arrays ID in it. So in this case, we could use the some other annotator DB SNP to get the arrays ID, and then we could query our database not with the chromosome position alt and ref, but with the arrays ID. Then we will have a column with weights in it, and then in different piece of Python code that will represent our reporter, we need to get weights from our PRS column and from the zygosity column, and then we could make weighted sum, and it will be our PRS value. So, and let me explain some our next steps that could be interesting for you in the way that you could use OpenCravat in several different ways. So first, if you work with PRS, sometimes you have a lack of information how you could interpret PRS values. And in this case, at least you could have percentiles and see how unique your data is for a particular genome. And for this purpose, we plan to use the GNOME-AD database. It have frequencies for the 8 populations and global frequency, and we could just have a generator script that would from one side get the PRS data, and from the other side would have frequencies for the particular SNPs, and this is enough to sample the data, and then we will have a normal distribution, and having sigma and mean, it's enough to have to calculate percentiles and all other information that we want. And we could go even further, and it's our next step. After the next step, we could identify a population of particular genome, and in this case, again, the GnomeAd database will help us. So in this case, we could use a Bayesian rule. So first, we will have a uniform distribution for all 8 population, and go from one SNP to another. We will update our prior probabilities. So for example, for first SNP here, as the prior probability, we will have 1/8 as a uniform distribution. And for example, we want to know the African frequency. So we would get the data about the African frequency and put it here is a probability of getting this SNP given we have African probability, and we will divide it out at the global frequency. On the next step, with the next SNP, we will take this probability as our result from the previous step and plug it as our prior probability, and get the P given A and PP from our database for the next SNP, and we could proceed this process for the each population until we will have a probability distribution for this particular genome. So in this way, we could tell whether you're African, American, Swedish, or someone else. So this is our plans, and we will investigate these possibilities with OpenCravat. Thank you, and now